to everyone here and to everyone joining us via Zoom webinar as well. I'd like to introduce our keynote discussion for tonight. I'm really excited to introduce a wonderful panel. Last Thursday, I was reading the New York Times and happened to come across a pretty interesting article hidden back on page 18, but I think particularly relevant to the discussion tonight. From no hope to potential cure for deadly blood cancer, study shows. And just to read a couple of excerpts from this, talking about the patients. They had gone through a series of treatments, each of which controlled their disease for a while, but then it came back, as it always does. But they entered a clinical trial of a new targeted immunotherapy. And what happened? A third of the patients responded so well that they got what seems to be an astonishing reprieve. Interesting article, but the same thing is happening today in our disease. And so with that, with this whole notion of targeted immunotherapy, I'd like to introduce our panel here. So our own Mark Firth, Chief Science Officer, Fibrolamellar Cancer Foundation, Juliane Walz, joining us from Tübingen, Germany. Molly Hones, a member of the FLC patient community who's experienced one of these targeted immunotherapy clinical trials firsthand. And last but not least, Mark Yarchoan joining us from Johns Hopkins. So with that, I'd like to turn things over to Mark to just key off a general discussion of what's happening in our disease in this arena.
Thanks, Kurt. And boy, welcome to all of you. I wish I could just go around and tell each of you how much I care and love what you're doing for this community, because it's fantastic. Tonight, we're not going to have a formal scientific presentation. There'll be plenty of that tomorrow and Wednesday. But I want to talk really about where we are on exactly what Kurt talked about. And I can say, when I joined the foundation almost 7 years ago, my dream was, first slide, following the words of the first great drug hunter, Paul Ehrlich, many of you know about. We need a magic bullet for this disease. It was a term he introduced well over a century ago. That idea of a magic bullet, today we call it targeted therapy. And it was obvious when I came in to think about, is there a way to do targeted immunotherapy? Because as John Scott had told me the first time I spoke to him, hey, you need normal protein kinase A2 if you want to live. And I was incredibly fortunate in the first few months of being with the foundation to learn about two groups that were working on exactly that problem and had already been making progress, and found out that very similar work had been going on as well in Tübingen, which we learned about when Juliane's team published an important paper 3 years ago. So it's no longer a movie being shown with Edgar G. Robinson in 1940. This is real-world science today. And we're not dealing with salvarsan for syphilis. We're dealing with cancer. And we're dealing with cancer in young people for whom there's no other solution. So I'd like to just give you kind of the big picture view of the progress that's been made on targeted immunotherapy for this cancer over the last few years, and get a little view at the end of some of the future steps that are certainly already on the blackboard or underway. And finally, to also make sure that you hear the voice of a patient who's been directly involved, not only as a member of a clinical trial cohort, but as somebody who's spoken with a lot of other patients, who's been willing to be public about her experience. So I'm going to ask Molly if she'll let all of you, who are mostly lab rats in the room, learn a little bit more about what the real world of the patient is like. I know there are a lot of clinicians and clinician scientists here who know it, but maybe even not from that deep first-person perspective that we'll hear about from Molly. So let's start now. Next slide. And you all know this. There's the thing we want to target, that DNA JB1 PRKCA protein. Let's just call it DP for the rest of the evening. And there was a notion that the junction between the D and P parts might be a targetable new antigen, a neoantigen. And how could one go after that? And that's the story you're going to hear tonight about at least one way to go after it by vaccinating against it with a peptide vaccine. And I'm going to ask Juliane to talk about how her department, which is actually named for peptide-based immunotherapy, functions in how that got into the story of fibrolamellar and their first clinical success, which was reported 3 years ago.
Thank you, Mark. Yes, maybe not all of you know Tübingen. Therefore, I brought this little map that shows you where Tübingen is located. And it's quite easy because there are two really famous locations near. It's 1 hour away from the Black Forest and half an hour away from Mercedes-Benz. And it's also one of the oldest university towns in Germany. And it's also really famous for immunology and immunotherapy. As Hans-Georg Rammensee worked there for nearly 20 years, and he is the one who discovered the binding of HLA and peptides, which is the central or the key component of tumor cells and all cells in the human body to allow the immune system to decide if a cell is foreign or not malignant or not infected. And yeah, in the lab of Hans-Georg Rammensee, I did my MD work, and I was from this time on really on this topic of cancer immunotherapy, and I continued to do my work there. And finally, I built up my own department, which is the Department of Peptide-Based Immunotherapy. And maybe you can just switch to the next slide or to the next. Yes, so this is just our team, and the department is a very, very specific structure that I think it's really unique because it's not only a research department. It's also a department that has the ability to produce peptide vaccines in clinical quality, so in GMP quality, so we can produce these vaccines for clinical trial to treat patients. And we have also a dedicated clinical department, which are all the people on the right-hand side, which is just built at a university hospital to treat patients in clinical immunotherapy trials. So we have the whole way from bench to bedside within one department. And I have to admit, even I'm a hematologist and oncologist, I've never heard about fibrolamellar cancer before. But at one time point in our so-called molecular tumor board, where genetic alterations in cancer were discussed, I was approached by the Children's Hospital with a patient called Maya, and she had a fibrolamellar cancer. And we discussed her in this molecular tumor board, and there was this specific gene fusion identified. And we were asked, you're doing peptide vaccines, why can't you do a vaccine against this gene fusion? And I thought, okay, this is an interesting project. And I approached Jens, who is here today, who is the scientific head of our lab. And I told him, hey, this would be a nice project. Just look at this fusion. Could we do a vaccine? And it really all started with this one patient. And so we did the whole process characterizing this amino acid sequence that makes up the fusion. We proved that it's naturally presented on tumor cells. We showed that it's detected by the immune system, by T cells. And then we finally produced this peptide vaccine targeting the fusion. And we treated Maya, who was in an advanced stage. She had liver transplant, she had chemotherapy, she had multiple disease relapses, and she was put finally on a palliative track, only treated with a PARP inhibitor based on the genetic alterations that were found in this molecular tumor board, but the relapses went on. And after the last relapse, we applied 2 doses of our peptide vaccine, and we were able to induce a very, very strong T-cell response targeting this gene fusion. And this T-cell response is still there. It's now nearly 5 years after the second vaccination. And we have very fresh T-cell immune monitoring data that we will show tomorrow. And T-cell response is still stable, and Maya is still disease-free. So she was 15 years old when we started treatment. She's now 19 years old, and she finished school. She started her university training, and she went from a palliative care patient now to a young girl that is just living her life. So, and this was the start of the whole story. And now we have a clinical trial running to provide this kind of therapy to more patients like Maya. And the second trial was funded by the FCF and is initiated, and we hope to include the first patient in this month.
Thanks so much.
Thank you.
Yarchoan and I met in January of 2019. I was still very, very wet behind the ears in the role I have now. And Mark will tell you maybe a little bit about that encounter. What he didn't know at the time was that I'd already had a conversation just a few days earlier with Paul Thomas, who's sitting in the room. And what Paul told me convinced me that a magic bullet for this cancer actually was not an impossibility at all. And so I'll let Mark tell you the story of his approach to a very similar story with a peptide vaccine to that fusion junction.
Well, I'm going to talk again tomorrow, and tomorrow is the sort of science-heavy talk. So tonight's the fun talk, and I think the less I speak and the more Molly talks, the better. So I'll try to be quick. I trained as a medical oncologist and am focused on laboratory-based cancer immunotherapy, focused on liver cancers. And I saw my first patient with fibrolamellar cancer just when I came out of fellowship. And it just seemed like a cancer that I should be doing something for. It just seemed to me that there were all these people working on immunotherapy for other forms of liver cancer, but as far as I knew, because I didn't know the team in Germany, I didn't know that there was anyone else working on this problem. So I worked on trying to design a mouse model of fibrolamellar cancer where I would try to give a vaccine to see if the vaccine could slow down the growth in the mouse. And I worked at this, and I wanted to create just enough mouse data that I could convince someone that this made sense to do in a patient. But I was working on it. We had some technical issues. And then I went to the GI ASCO meeting in San Francisco in January of 2019, right before the world shut down. And I met with Mark Firth, and I really wanted to show him the mouse data, but I didn't actually have it. So I said, well, Mark, I have this idea. I have no data to back it up, but what do you think about this? Is this something that you guys would be interested in funding? And amazingly, the foundation gave just enough money to sort of push this along in a couple of patients and allow us to compete for more money afterwards. So next slide. And I think, Mark alluded to this, but the reason that the foundation felt comfortable with this is that they were aware that Paul Thomas at St. Jude had found T cells that recognize this fusion event in patients. And so it turns out that all this work that I was doing in the mice might not be needed because there was human data that I just wasn't really aware of. And so Mark actually connected Paul and myself, and I think we've had a very collaborative relationship since that time. So next slide. So, the other person that I just want to acknowledge is sitting in the audience, and that's Marina Beretti. So Marina was visiting us from Italy, and I don't know why anybody would leave the food and the piazza and everything that she left to come to Baltimore, Maryland, but she did. And she's been my clinical partner ever since. And, she worked with me on this trial when she was here visiting from Italy, and then chose to stay and redo fellowship, and then has continued to work with me ever since. And I honestly don't think I could do what I do now without her. This is the rest of our team, and it's not even a complete team picture here, but a clinical trial involves lots and lots of people. These are the true heroes of the trial that we're presenting, and the trial was combining a vaccine with checkpoint inhibitors for patients with FLC. We'll be showing the data tomorrow, but just a little teaser. So next slide. So this is the first patient that responded on the clinical trial. And this was amazing for me. It was great for the patient. But what many people don't know is that trials have futility built in. So if you don't see activity, you have to shut the trial down. And if this patient had not responded, we never would have seen what would have come afterwards. So I'll never forget the moment when I saw the scan for the first time. And this patient at the last data cutoff remains without active tumor now going on 5+ years, and actually came to us on hospice. So I will now turn it over to Molly.
Hi, I'm Molly Hannes. First of all, I want to say thank you for all of you here. Without you, I would not be here. Next slide, please. My journey started about 2 years ago, almost actually to this week. I was diagnosed as a stage 3 FLC patient after a successful liver resection surgery and splenectomy. Because of positive margins, I completed several rounds of radiation and radio chemo. At that point, I was no evidence of disease in November and decided to not seek adjuvant care because I felt like I needed to let my body heal and let my mind heal as well. Going through cancer treatment is really stressful, and I knew from the beginning it would be a marathon, not a sprint, especially given how many people experience recurrences. I had my first recurrence a year ago in May. It had spread to my lungs. And then in May 2024, incredibly, only 3 weeks it took to get through all the insurance approvals, I had driven out to Baltimore and I received my first peptide vaccine at Johns Hopkins. Throughout that summer and throughout treatment, I'm really excited to share that I've been able to maintain an extremely high quality of life and have had minimal side effects. In fact, my only side effect, and I can remember having calls with Dr. Veretti about this, was I had a little bit of itchiness on my legs after the first time swimming in a lake in like 5 years. Safe to say it was probably swimmer's itch and not the immunotherapy. Next slide, please. And then in September 2024, I'm proud to announce I was no evidence of disease and I'm currently experiencing remission. I've also had several Natera tests as well, and they're not even picking up circulating tumor blood in my DNA. So that is my path. And again, thank you.
Molly, could I ask you to really, partly for the benefit of patients and caregivers who are going to be listening in or who may look at the tape of this on the web, maybe you could drill a little more into the experience of being in a clinical trial. What did you actually have to do to participate? Were there barriers to it? Did you have any hesitation that clearly you overcame? But what was the sense of stepping off that cliff into the unknown?
So back before I was in remission in 2023, my oncologist and I were actually already discussing clinical trials. I've always been passionate about science and technology and what the future could bring. My uncle was diagnosed with lymphoma back in the early 2000s and was given only a couple of months to live and hopped on a trial. He's still with us today. I talked to my uncle Mike. He gave me some motivation, and I'm like, you know what? I want to contribute too, and maybe I'll get something in return as well. And from there, we just had that as a point of conversation in every appointment with my oncologist in Madison, just because I liked being, kept abreast with what's going on. But also, when we talked about the peptide vaccine trial at Johns Hopkins, it got brought up in conversation because it's genuinely a very exciting trial and a very exciting technology. So then, when I found out that my cancer had returned, you would think that that initial news would be very shocking, and it was, but I knew that there were options out there. and that if I pursued that magic bullet and my body was feeling good, let's do it. Why not? So from there, it was just a lot of communication between Johns Hopkins and then my team in Wisconsin. I think the biggest hurdle, honestly, was the waiting and trying to stay grounded, trusting in the science when there is minimal data, but the data sure looks really promising. And getting everybody else in my family on board of like, we're going to do this. We're going to take the time and the investment to go out to Baltimore. And it paid off.
Thank you. I think, and anybody who's been involved in translational research will swear by the importance of those folks who volunteer to participate in trials. They're the real heroes and heroines of this story. Let's give Molly a hand. I agree. Thank you. And part of the mission, I think, right now for some of us is to remind the patient community, if you have a disease for which there's not a known therapy that works, your best bet might actually be to go into that experimental realm, to try something new, especially if it's based on good, solid science. And one of the foundation's goals is to help bring as many of those opportunities as possible to the patient community in as short a period of time as possible. And I think that's, Marna and Chuck get a lot of credit for that because they help provide the money to do it. As Marna said, it's expensive, but it's worth it. With that, maybe I'll ask Juliane to just tell us a little more about the 2 clinical trials that are being carried out in Tübingen with the peptide vaccine, a little more of the detail and what the goals are.
Sure. So the first trial that is already running now for nearly 2 years, we started in patients with advanced fibrolamellar cancer, so non-surgically resectable or metastatic disease. And in this trial, we combined the peptide vaccine with an immune checkpoint inhibitor, atezolizumab, which is provided from a company called Roche. And patients start with the peptide vaccine, so they get 2 doses of the vaccine. And after 2 weeks after the first dose, we start the immune checkpoint inhibitor treatment every 4 weeks. It's a phase 1 clinical trial and will recruit 20 patients. The first 10 have already been recruited. And as a phase 1 trial, primary endpoints are safety and immunogenicity, so induction of T-cell responses. Secondary endpoints are, of course, then clinical responses. We assess these patients with CT scans every 8 to 12 weeks. And we really hope to see positive results. And we show some of these tomorrow in the session. And what we realized already when we started the trial, but more when I visited the patient summit last year and I presented the data in the trial. And I think 4 families approached me and asked me, why can't I participate in the trial when I have no tumor at the moment? There were a lot of patients that were tumor-free or were on a continuous treatment. And they could not in the trial because there was no measurable tumor lesion. And they asked me, can't you do a trial to provide the vaccine also in an adjuvant setting? And this was the start point for us to say, okay, we want to evaluate our vaccine as a single agent, so without immune checkpoint inhibitors, in an adjuvant setting, so in all FLC patients that achieve a remission, independent if it's after surgery, after radiation, after any systemic treatment. And we set up this trial. Again, it's a phase 1 trial. Again, we will recruit 20 patients. And we initiated that trial in the end of May. And we just received the next delivery of vaccine product, and we are now ready to recruit the first patients in this adjuvant trial. And maybe one thing to mention, the German regulatory authorities are very strict regarding children, even if we believe this vaccine is safe for children. So Maya was a child when we treated her. But both trials are so far only open for patients older than 18. However, the regulatory authorities envisage us to include children if we provide safety and first immunogenicity data. So we hope to soon also recruit children.
Thank you so much. I can tell you the foundation is just thrilled to have received the grant application for that adjuvant trial. Some of the folks who helped us read that grant are nodding, and it was a case where there was just a tremendous outpouring of support, and we're very glad that that's now really ready to recruit patients. It's fantastic. Mark, I wanted to turn back to you if I could, and you've already talked about the original peptide vaccine trial, but your group's also been focused on other approaches, and specifically the question of why only some of the patients who show an immune response are showing these really fantastic clinical responses, and also on other potential ways to take advantage of that target and the immune system that may be better for some patients than a vaccine. So could you just kind of give us a little bit of a forward snapshot and snapshot of some of the stuff you're going to talk about tomorrow?
Yeah, sure. So, we've done a lot of work trying to understand why it is that we give the same vaccine to this group of patients. Some of them respond, some of them don't. There's a group of patients who you give the vaccine to who just don't mount responses against the vaccine target. But the majority of patients actually did mount a response to the vaccine target. And so I think that's the most perplexing for us. You mount a T cell response against a target. That target is on the cancer cell, and yet the patient didn't respond. And why is that? Well, clearly these tumors have a program that causes immunosuppression. And we'd like to sort of figure out what that program is. One of the thoughts that we have in our group, and Dr. Beretti will be presenting this tomorrow, is that these tumors have abnormal metabolism, and that metabolic profile can actually be very toxic to T cells. And Dr. Beretti will be leading a trial adding a glutamine antagonist to see if we can rescue some of these responses and eventually hope to add that to our current vaccine trial. The other thing is that we do think that the patients who respond probably have better T cell responses against the target. Not all T cells are created the same. And one of the things that we're actively working on, and Paul Thomas will talk about this tomorrow, we're working together on this, is to really take the T cell program from the patients who do really well, like Molly, and then actually use that program to reprogram T cells for other patients. So we can skip the vaccine altogether and just go straight into what's called an adoptive cell therapy. So that's another thing that we're working on. I will say that I think that this adjuvant trial that's happening is really elegant, and I think that's where our field needs to go. Clearly, vaccines work better the earlier you use them. And one of the things that we've noticed is that a lot of the patients who come to our trials come very, very late in this country. Basically when they really are told that there are no other options, and that's just not the ideal time to do a vaccine therapy. So, we've looked at our data, and every single patient who responded had never seen GemOx lenvatinib. So, I think it's hard to tell a patient to do a totally experimental therapy first, but I do think based on the mechanism of action that the earlier you give these vaccines, the better, and hopefully our field can move more in that direction.
Thanks, Mark. I don't want to keep people very much longer, but I actually wanted to give Molly the last word. Molly, any thoughts that you'd like to communicate to your friends and colleagues both here and outside?
Sure. Oh, but before we start talking about the future, I want to take everybody back about 30 years ago. It's the mid-90s. Some of you might have been in school. Some of you have already been practicing medicine. I was in kindergarten. And I had a classmate who also happened to be named Molly. Molly did not make it to first grade because she passed away from pediatric leukemia. My class, all 25 of us, until we were seniors in high school, participated in the American Cancer Society's Relay for Life, raising money for research and for cancer services in our area. And every year, we heard stories about cure, about new therapies, about how people weren't passing away from cancer like they're used to. People were getting hopeful. I carried that hope with me, even when I was diagnosed with cancer. And one of the things that I reflected upon a lot over the past 2 years is cancer takes away a lot of things from you as a patient, especially a young person. And when I found out that I could be doing something more than just raising money and walking laps, that I could be contributing to bringing that cure that people talked about that might be a pipe dream. No, that's coming sooner than we think. And it's because of people like you. It's because of folks who volunteered for trials ahead of me. And I think before every appointment, I dedicate each treatment to somebody. And I would just like to say, today and just offering it up, we're here for everybody who came before, and we're here for everybody who might be diagnosed in the future. But one thing is for certain, that cure is coming, and it's all because of the great teamwork that has been put together by FCF. Thank you.
Thank you, Molly. For me, the patient with leukemia I met when I was a teenager was named Jenny. She was 5. And I think all of us have probably had some experience like that, but it is what keeps us going. And I think, Marna, you and I are now probably about the same ballpark age.
Old.
You said it. I think we're going to do a lot about this within our lifetime, yours and mine. And we've seen the beginning of it. We'll hear a lot more of the data tomorrow. We'll hear about a lot of the great work that's going on targeted therapy, on metabolism, et cetera, et cetera. Have a good night and be ready for a lot of fun tomorrow and Wednesday.